itself. Just like you said, the mind is kind of we call it story narrative. There's a simulation in our, so our mind is essentially a simulation.
And usually I try to...
The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to copy the A's, C's, T's, and G's of the gene. You are watching this process called transcription in real time. It's happening right now in almost every cell in your body. The job of this mRNA is to carry the gene's message from the DNA out of the nucleus to a ribosome for production of the particular protein that this gene codes for. There can be several million ribosomes in a typical eukaryotic cell. These complex catalytic machines use the mRNA copy of the genetic information to assemble amino acid building blocks into the three-dimensional proteins that are essential for life. Let's see how it works. The ribosome is composed of one large and one small subunit that assemble around the messenger RNA, which then passes through the ribosome like a computer tape. The amino acid building blocks, that's the small glowing red molecules, are carried into the ribosome attached to specific transfer RNAs. That's the larger green molecules, also referred to as tRNA. The small subunit of the ribosome positions the mRNA so that it can be read in groups of 3 letters known as a codon. Each codon on the mRNA matches a corresponding anticodon on the base of a transfer RNA molecule. The larger subunit of the ribosome removes each amino acid and joins it onto the growing protein chain. As the mRNA is ratcheted through the ribosome, the mRNA sequence is translated into an amino acid sequence. There are 3 locations inside the ribosome, designated the A site, the P site, and the E site. The addition of each amino acid is a 3-step cycle. First, the tRNA enters the ribosome at the A site and is tested for a codon-anticodon match with the mRNA. Next, provided there is a correct match, the tRNA is shifted to the P site, and the amino acid it carries is added to the end of the amino acid chain. The mRNA is also ratcheted on 3 nucleotides, or 1 codon. Thirdly, the spent tRNA is moved to the E site, and then ejected from the ribosome to be recycled. Using computer animation based on molecular research, we are now able to see how DNA is actually copied in living cells. You are looking at an assembly line of amazing miniature biochemical machines that are pulling apart the DNA double helix and cranking out a copy of each strand. The DNA to be copied enters the production line from bottom left. The whirling blue molecular machine is called helicase. It spins the DNA as fast as a jet engine as it unwinds the double helix into 2 strands. One strand is copied continuously and can be seen spooling off to the right. Things are not so simple for the other strand because it must be copied backwards. It is drawn out repeatedly in loops and copied one section at a time. The end result is 2 new DNA molecules. we'll see the remarkable way our DNA is tightly packed up so that 6 feet of this long molecule fits into the microscopic nucleus of every cell. The process starts when DNA is wrapped around special protein molecules called histones. The combined loop of DNA and protein is called a nucleosome. Next, the nucleosomes are packaged into a thread. The end result is a fiber known as chromatin. This fiber is then looped and coiled yet again. To the familiar shapes known as chromosomes, which can be seen in the nucleus of dividing cells. Chromosomes are not always present. They form around the time cells divide when the 2 copies of the cell's DNA need to be separated.
DNA in each one of your cells is just 2 nanometers across, but around 30 to 40 million nanometers long. To keep DNA organized and regulate access to the genetic code, it is wrapped around structural proteins to form nucleosomes, which are then further organized into fibers called chromatin. The chromatin fibers are packaged into gigantic molecular structures called chromosomes. We are now pulling away from a chromosome, out through a nuclear pore, which is a gateway through the nuclear membrane. We are zooming back to reveal a cell about to undergo mitosis. The nuclear membrane breaks down, releasing the chromosomes. Chromosomes are arranged in the middle of the cell before being pulled apart, with identical sets of DNA going to each side. The cell then splits down the middle, creating 2 new daughter cells. This visualization isolates a single chromosome at mitosis. The chromosome contains 2 identical copies of DNA. Each DNA molecule is packaged up into one of the sausage-shaped chromatids. Sticking out from either side are microtubule fibers from the mitotic spindle used for orienting and guiding the chromosomes to their correct positions. The red region is the kinetochore, which works as the interface between the microtubules and the chromosomes. The kinetochore is one of the largest and most complex molecular mechanisms inside living cells, with multiple functions to perform. The kinetochore is central to the movement of the chromosomes and is able to hang on to the dynamic ends of the microtubules. It is also a mechanical tension sensing system, which is able to feel if everything is correctly attached and positioned. It is also a stop signal broadcasting system, sending out chemical signals to the rest of the cell about whether it is ready to undergo separation of the chromosomes. This kinetochore has sensed that the tension is just right and the chromosome is correctly positioned and attached. The last little bit remaining red is the stop signal broadcasting system. It is carried away by a dynein motor, walking down the microtubule away from the kinetochore. These are molecular motors involved with a directed transport along the mitotic spindle. The orange proteins walking to the left are kinesins, molecular motors carrying proteins on their back. Striding from the kinetochore on long molecular legs are dynein, carrying the stop signal broadcasting system.
Of the purpose that the mind has. It's a tool for modeling, right? We are not actually monkeys. We are side effects of the regulation needs of monkeys. And what the monkey has to regulate is the relationship of an organism to an outside world that is in large part also consisting of other organisms. And as a result, it basically has regulation targets that it tries to get to. These regulation targets start with priors. They're basically like unconditional reflexes that we are more or less born with. And then we can reverse engineer them to make them more consistent. And then we get more detailed models about how the world works and how to interact with it. And so these priors that you commit to are largely target values that our needs should approach, set points. And this deviation to the set point creates some urge, some tension. And we find ourselves living inside of feedback loops, right? Consciousness emerges over dimensions of disagreements with the universe. Things where you care, where things are not the way they should be, where you need to regulate. And so in some sense, the self is the result of all the identifications that you're having. And identification is the regulation target that you're committing to. It's a dimension that you care about, that you think is important. And this is also what locks you in. If you let go of these commitments, of these identifications, you get free. There's nothing that you have to do anymore. And if you let go of all of them, you're completely free and you can enter nirvana because you're done.
And actually, this is a good time to pause and say thank you to a friend of mine, Gustav Soderstrom, who introduced me to your work. I wanted to give him a shout out. He's a brilliant guy, and I think the AI community is actually quite amazing, and Gustav is a good representative of that. You are as well. So I'm glad, first of all, I'm glad the internet exists, and YouTube exists, where I can watch your talks and then get to your book and study your writing and think about, that's amazing. Okay, but you've kind of described sort of this emergent phenomenon of consciousness from the simulation. So what about the hard problem of consciousness? Can you just linger on it? Like, why does it still feel like, I understand you're kind of, the self is an important part of the simulation, but why does the simulation feel like something?
So if you look at a book by, say, George R.R. Martin, where the characters have plausible psychology, and they stand on a hill because they want to conquer the city below the hill, and they've done it, and they look at the color of the sky, and they are apprehensive and feel empowered and all these things. Why do they have these emotions? It's because it's written into the story, right? And it's written into the story because it's an adequate model of the person that predicts what they're going to do next. And the same thing is true for us. So it's basically a story that our brain is writing. It's not written in words, it's written in perceptual content, basically multimedia content. And it's a model of what the person would feel if it existed. So it's a virtual person. And you and me happen to be this virtual person. So this virtual person gets access to the language center and talks about the sky being blue. And this is us.
But hold on a second. Do I exist in your simulation?
You do exist in an almost similar way as me. So there are internal states that are less accessible for me that you have, and so on. And my model might not be completely adequate. There are also things that I might perceive about you that you don't perceive. But in some sense, both you and me are some puppets, two puppets that enact this play in my mind. And I identify with one of them because I can control one of the puppets directly. And with the other one, I can create things in between. So for instance, we can go in an interaction that even leads to a coupling, to a feedback loop. So we can think things together in a certain way or feel things together. But this coupling is itself not a physical phenomenon. It's entirely a software phenomenon. It's a result of two different implementations interacting with each other.
So that's interesting. So are you suggesting, I like the way you think about it, is the entirety of existence a simulation, and we're kind of, each mind is a little sub-simulation? That, like, why doesn't your mind have access to my mind's full state? Like,
for the same reason that my mind doesn't have access to its own full state.
So what,
there is no trick involved. So basically, when I know something about myself, it's because I made a model. So one part of your brain is tasked with modeling what other parts of your brain are doing.
Yes, but there seems to be an incredible consistency about this world in the physical sense, that there's repeatable experiments and so on.
Yeah.
How does that fit into our silly Center of Apes simulation of the world? So why is it so why is everything so repeatable? And not everything. There's a lot of fundamental physics experiments that are repeatable for a long time, all over the place, and so on. Laws of physics. How does that fit in, you think?
It seems that the parts of the world that are not deterministic are not long-lived. So if you build a system, any kind of automaton, so if you build simulations of something, you'll notice that the phenomena that endure are those that give rise to stable dynamics. So basically, if you see anything that is complex in the world, it's the result of usually of some control, of some feedback that keeps it stable around certain attractors. And the things that are not stable, that don't give rise to certain harmonic patterns and so on, they tend to get weeded out over time. So if we are in a region of the universe that sustains complexity, which is required to implement minds like ours, this is going to be a region of the universe that is very tightly controlled and controllable. So it's going to have lots of interesting symmetries and also symmetry breaks that allow to the creation of structure.
But they exist where? So there's such an interesting idea that our mind is simulation that's constructing the narrative. My question is, where I can watch your talks and then get to your book and study your writing and think about, that's amazing. Okay, but you've kind of describing sort of this emergent phenomena of consciousness from the simulation. So what about the hard problem of consciousness? Can you just linger on it? Like, why does it still feel like I understand you're kind of the self